My name is Evangelina López de Maturana. I am staff scientist in the Genetic Molecular Epidemiology Group led by Nuria Malats in the Spanish National Cancer Research Center in Madrid. And I've been working in the genetic epidemiology field of pancreatic cancer since 2016. I've been working mainly on the identification of high-risk populations by integrating multimodal multi-omics data, as well as in the identification of alternative biomarkers for early detection. In the tertiary prevention field, I've been also involved in the identification of prognostic signatures by also integrating multimodal genomics data by applying machine learning algorithms. So I share common interest with this platform. I think that is a very good resource to improve the communication between not only researchers and clinicians, but also with patients groups in order to increase the awareness of this disease. So I think that it has provided me with a wider vision of why research is important in this disease and how the crosstalk between different people may help us to move forward in the advancements of the knowledge of this disease. So I think that there are several. First, the lack of tools to identify these high-risk populations of developing sporadic pancreatic cancer is a very important challenge, because refining this high-risk population would help us to apply screening tools in order to advance the diagnosis of this disease. Also, another important challenge in this disease is the lack of effective treatment. So I think that it's important, as I said before, like to join efforts in this regard, because it's a very low incidence tumor. And I think that improving the communication, not only between researchers and clinicians, and also taking into account patients groups would help in this regard. So I think that there have been several advancements. The first one is that so there are increasing amounts of multimodal data that have been collected in a large amount of people. So this would help us to redefining better these high-risk populations in order to apply screening tools in a more cost-effective way. Also, liquid biopsy is a very promising tool, not only for early diagnosis, but also for tailoring treatment and also for detecting recurrence at earlier stages. And also the development of drugs that target specific KRAS mutations also are important and have shown important or promising results in recent clinical trials. So I think that we can be optimistic in this way. So I would like to thank you all for your generosity and your willingness to participate in research projects whenever it's possible, because joining efforts and is crucial to advance in this regard. So your participation is very, very much important to advance in the knowledge of this disease, to diminish the incidence, and also to diminish the mortality associated to this disease. So thank you.